Imagine having a word processor for human DNA.
Oh, that's a wild thought.
Right. Like you aren't just fixing a single spelling mistake. You're highlighting entire massive chapters of a genetic code, copying them, and pasting them flawlessly into another book.
And doing it without tearing the paper or, messing up the formatting.
Exactly. That level of absolute control. Welcome to the deep dive. Today, we're looking at a groundbreaking paper that brings that exact concept out of science fiction and into the laboratory.
Yeah, it's really exciting stuff.
We are digging into a new genetic engineering strategy called QuadPE. That stands for quadruple pegRNA.
Right, quadruple, 4 parts.
Our mission for this deep dive is to unpack exactly how this 4-part molecular machine works, why it overcomes the frustrating limitations of our current genetic tools, and how it could eventually pave the way for universal cures to complex genetic disorders.
It really is a game changer for the field.
Okay, let's unpack this. Where do we even start with the current state of genetic copy-pasting?
Well, we have to start with the fundamental problem we're trying to solve. Most genetic diseases are caused by specific sequence mutations.
Like literal typos in your DNA.
Exactly. Now, current tools like base editors and prime editors, they are brilliant at fixing those tiny single-letter typos.
Okay, so what's the issue then?
The clinical application is severely bottlenecked by the sheer diversity of those mutations. You might have a disease, let's say cystic fibrosis, caused by a typo on page 5 of your genetic code.
Right.
But someone else has the exact same disease, but their typo is on page 50.
So, if I'm understanding that right, we'd have to design a custom, highly specific therapy for every single patient's individual typo.
Yes, you would.
That sounds incredibly slow. And expensive.
It is both, which is why the holy grail of genetic engineering isn't typo correction, it's targeted gene insertion.
Ah, okay. So instead of hunting down individual mistakes, you just replace the entire faulty gene.
Right. You take the whole chapter out and replace it with a completely healthy one. It's a universal fix.
It wouldn't matter where your specific mutation is.
Exactly. But traditional methods for pasting in whole chapters have fallen painfully short.
Let's break down why they fail. Because scientists have been trying to paste DNA into the genome for a while now, haven't they?
They have, yeah. The oldest methods rely on creating what we call a double-stranded break, or DSB.
Oh, the molecular scissors?
Right, like the standard CRISPR-Cas9 system. You basically take those scissors and you snip completely through both strands of the DNA helix.
Which sounds pretty violent for a cell.
It is. The cell naturally panics. It sees its DNA severed in half and tries to glue the ends back together using a biological emergency response pathway.
And that's called non-homologous end joining, right? NHEJ.
Spot on. NHEJ.
That sounds like fixing a severed wire with duct tape in the dark.
That is essentially what the cell is doing. It's incredibly messy.
Yeah.
This pathway is prone to creating random insertions or deletions, which geneticists call indels.
Indels, okay.
Yeah. And if you are trying to slip a new piece of DNA into that break while the cell is panicking, the efficiency is just dismal. You're typically looking at a success rate in the range of just 1 to 5%.
Wow. Okay, so snipping entirely through both strands of the DNA is out. It's way too chaotic.
Way too chaotic.
Yeah. But I've seen something else mentioned in the literature, homology-directed repair, HDR. Does that solve the chaos?
Homology-directed repair is a natural cellular process that is much more precise. It uses a donor template to perfectly fix the break, mashing the sequences up.
It was better than duct tape.
Much better. But here is the fatal flaw with HDR. It largely requires the cell to be actively dividing for the pathway to turn on.
Oh, I see.
Yeah, if you want to fix a genetic disease in tissues where cells aren't dividing, which includes many clinically critical areas like the brain or the heart, HDR is effectively useless.
Wait, so if the process only works in dividing cells, is that like trying to rebuild a car engine, but you are only allowed to do it while the car is actively speeding down the highway?
That's a really great way to visualize it. If the car is parked in the garage, which is what non-dividing cells are basically doing, the repair mechanism shuts down. You can't make the swap.
Man, that's frustrating. Let's say I'm looking at a neurodegenerative disease like Huntington's. Brain cells don't divide.
Exactly. So HDR won't help you there. Because of that severe limitation, scientists developed a newer wave of tools using integrases and transposases.
Right, I've seen those. Systems with names like PASSAGE, paste, and cast.
Yeah, those are the ones.
So these are the newer generation of tools. Do they let us fix the engine while the car is parked?
They do, but they come with incredibly heavy baggage. These integrase methods require a complex two-step process.
Okay, 2 steps. Like what?
First, you have to edit the genome just to install a specific landing pad sequence. Then, you use a massive integrase protein to drop the new DNA onto that pad.
Landing pad, okay.
But because these protein machines are so physically huge, delivering them into the nucleus of a cell is an absolute nightmare.
Just getting them through the door is a problem.
Exactly. Furthermore, their insertion efficiency drops sharply the larger your new DNA payload gets.
I see. But hold on. The paper we're looking at today builds on prime editing.
It does.
If prime editing is so precise and it doesn't require cutting both strands of the DNA, why can't we just prime edit a really long piece of healthy DNA directly into the genome?
It's a logical question, but it comes down to how prime editing physically works. Traditional prime editing relies on an enzyme called reverse transcriptase.
Reverse transcriptase, okay.
It essentially writes the new DNA sequence right there on the spot, filling in a tiny gap. It works beautifully for short sequences.
Yeah.
But reverse transcriptase is an enzyme that tires out quickly. Once your new sequence exceeds about 300 base pairs, it basically runs out of steam.
300 base pairs, that's tiny.
Very tiny. The efficiency just plummets to 0 after that.
Okay, let's just do a quick recap so we can keep all this straight before we move into the actual solution.
Good idea.
So current tools fix small typos, but fail at pasting whole paragraphs. Double-strand breaks, they're too messy, giving us that 1 to 5% efficiency.
Right, the duct tape method.
Though we have HDR, which requires dividing cells, and prime editing hits a hard wall at a size limit of just 300 base pairs.
You've outlined the exact barrier the field has been slamming into.
So here's a question for you listening right now. Think about the cells in your brain right now. They aren't dividing. How would you fix a genetic typo in a closed book?
It's the multi-billion-dollar question. And that seemingly impossible barrier is exactly what the researchers who built QuadPE set out to shatter.
So let's talk about how they did it. Enter QuadPE.
The solution is as elegant as it is complex. QuadPE stands for quadruple pegRNA.
Let's break that down. A pegRNA.
Right, a prime editing guide RNA. It is essentially a molecule that serves 2 functions at once. It acts as a GPS beacon to find the exact right spot in the DNA.
Okay, GPS.
And it also carries a tiny blueprint giving instructions on what to do once it gets there. To understand QuadPE, we need to visualize a highly coordinated molecular 4-way handshake.
A 4-way handshake, okay.
The system relies on 4 of these engineered pegRNAs operating simultaneously.
Okay, 4 independent guides all working at once. That sounds complicated. What exactly is each pair doing?
Let's split them up to make it clearer. 2 of these pegRNAs target the genomic integration site.
So that is the exact spot in the patient's DNA where you want to paste the new gene.
Exactly. They guide a modified Cas9 protein to nick the DNA. But here's the genius part. They don't just nick it.
What else do they do?
They are programmed to synthesize a very specific 30-nucleotide overhang, which we call a 3' flap.
A 3' flap. Let's unpack that. I'm imagining a little piece of string.
Yeah, that works.
Think of the 3' end just as the tail end of the DNA strand. So it's like a little tail of single-stranded DNA hanging off the main double-stranded helix.
That's a perfect image. So now you have 2 of these tails, or flaps, hanging off the patient's genome.
Okay, got it.
Meanwhile, your other 2 pegRNAs are targeting your exogenous donor DNA. That's the brand new healthy gene you want to insert.
Right.
They operate on that donor DNA to create 2 matching 3' flaps.
Okay, I think I see where this is going. It's almost like a space docking sequence.
Oh, I love that analogy.
Yeah, you've got the International Space Station representing the human genome and an approaching shuttle representing the new donor DNA.
Right.
Instead of just ramming into each other, both the station and the shuttle extend these perfectly matching 30-foot docking arms. Those are our flaps.
You're getting the exact idea. And because the sequence of chemical letters on the genome's flaps perfectly match the letters on the donor DNA's flaps, they naturally seek each other out.
Because of sequence homology.
Exactly. The scientific term is that they anneal.
Anneal.
Well,
so they basically just zip together chemically.
They chemically zip together. And by doing so, they guide the massive new DNA payload securely into place without ever needing to snap the whole space station in half with a double-stranded break.
That's incredible.
The flaps do all the physical alignment. But the researchers didn't stop at just proving this works. They had to test two distinct architectural designs for how those initial genomic flaps are physically oriented.
Right, the PAM configurations. They refer to these as PAM out and PAM in.
Yes.
Let's clarify what PAM is for a second. That's essentially a small molecular signpost that tells the editing machinery where to attach, correct?
That's a great way to describe it. Protospacer adjacent motif, or PAM, is just the anchor point. So let's look at the GPM out design first.
Okay, PAM out.
In this configuration, the nicking sites on the genome are positioned so they face outward, away from each other.
Outward.
When those flaps form and the donor DNA zips into place, it promotes a local unwinding of the main genomic DNA in between those two nicks.
And what happens to that unwound section?
The cell's natural machinery comes in and fills the single-stranded gaps, which actually results in the duplication of the original genomic sequence on either side of the newly inserted gene.
Wait, it leaves a duplicate of the original DNA behind? What is the benefit of that?
Pure efficiency. Unwinding the DNA that way is incredibly natural for the cell, so it strongly promotes the insertion. It is highly, highly efficient.
Okay, so if we need a memory trick here, a PAM out means outstanding efficiency, but it pushes out a duplicate.
That's a good way to remember it.
So what if I'm a researcher and I don't want any duplication? What if I'm looking to completely erase a faulty gene without a trace and seamlessly replace it?
Then you would utilize the GPM in configuration. Here, those nicking sites face inward.
Okay, inward this time.
When the donor DNA zips into place, it actually forces the intervening genomic DNA, the old faulty stuff, to be displaced as loose 5' flaps.
Think of the 5' end as the head of the strand.
Right.
So the old DNA just gets pushed out of the way. What happens to it?
The cell recognizes those displaced loose ends as trash. It sends in enzymes called nucleases to naturally degrade and recycle them.
Oh, wow.
Yeah, the result is a perfect deletion of the old sequence, entirely replaced by your new sequence.
That's a brilliant mechanism. It's like an invisible eraser.
Yeah.
But I assume there's a catch. The paper mentions that PAM is less efficient than PAM out, right?
Considerably less efficient, yeah. PAM out is your heavy lifter for pure insertion. PAM is your surgical eraser, but it's physically harder for the cell to pull off.
Makes sense.
Now, dialing in either of these systems required immense optimization. They had to find the thermodynamic Goldilocks zone for those flaps we talked about.
The 30 nucleotide flaps. Why does the length of the flap matter so much?
It's all about physical stability. If the flaps were only, say, 10 nucleotides long, the editing completely failed.
Just not enough grip.
Exactly. There just aren't enough hydrogen bonds in 10 nucleotides to hold massive DNA strands together. They just float apart before they can zip up.
And if they're too long?
If you make them too long, like 50 nucleotides, the strands tangle up on themselves. They found that exactly 30 nucleotides, with about 50% GC content, meaning an even balance of guanine and cytosine bases, was the thermodynamic sweet spot.
The Goldilocks zone.
Exactly. Robust insertion every time.
And they didn't just optimize the flaps, they also spent a lot of time optimizing the physical shape of the donor DNA itself.
It did.
The paper compares linear DNA donors, which they called LV4, to circular DNA donors, called CV6.
And when it came to carrying massive DNA sequences, the circular CV6 donors won by an absolute landslide.
Why would a circle perform better than a straight line?
Because the inside of a living cell is a highly defensive, hostile environment. Cells have immune enzymes called exonucleases.
Exonucleases.
Think of them like little Pac-Men. Their sole job is to chew up loose, exposed DNA ends, because loose DNA usually implies an invading virus.
Oh, I see. And linear DNA has exposed ends.
Precisely. So the larger the linear piece, the longer it takes to integrate, and the more likely it gets chewed up and destroyed by those Pac-Men.
But circular DNA.
Circular DNA has no ends.
Yeah.
It's a continuous closed loop. The cell's defenses can't bite into it, allowing it to safely deliver massive payloads.
Okay, let's do another quick recap of all that machinery. We've got a system using 4 RNA guides. It creates matching 30 nucleotide flaps that chemically zip together.
Right.
We know PAM out duplicates DNA for high efficiency, PAM in cleanly deletes it, and circular DNA avoids the cellular Pac-Man.
That's a perfect summary.
So listener, think about this. If you were designing a gene therapy, would you prefer the highly efficient PAM out that leaves a slight genomic duplication, or the PAM in that perfectly replaces the sequence but is way harder to pull off?
It's a tough choice for bioengineers.
So we have a highly optimized tool in theory. But theory is one thing. I want to know how this actually performed in the lab. How does this system benchmark against the heavyweights of the industry?
Prepare to be surprised. Remember when I said prime editing falls off a cliff after just 300 base pairs?
The reverse transcriptase gets tired.
Right. Well, QuadPE successfully integrated DNA fragments starting at 1.6 kilobases, which is already a massive leap, all the way up to 26 kilobases.
26,000 base pairs. That is an absolute unit of DNA. We aren't talking about genes anymore.
Exactly. It is an entire genetic circuit. And the crucial metric isn't just that it can do it, it's how well it does it. Let's look at the head-to-head stats from the paper.
Let's hear them.
The researchers tested QuadPE with a 9.5 kilobase fragment at major therapeutic locations in the genome-specific addresses known as VGFA, HEK3, and TREC. QuadPE achieved around 40% efficiency.
40% for nearly 10,000 base pairs. That's staggering. How does that compare to those bulky, complex integrase models we talked about earlier, the pasty and Cas systems?
It completely outclassed them. When inserting these large payloads, QuadPE was 11 times more efficient than Passage, 61 times more efficient than pasty, and 12 times more efficient than Cast.
61 times better than paste. Wow.
Yeah. To prove the inserted DNA was actually functional, they even dropped in a 3.5 kilobase sequence that restored something called GFP expression.
GFP, green fluorescent protein, right.
Exactly. Essentially, they made the humor cells glow green under a microscope, physically proving the inserted gene was alive, integrated, and actively working.
Okay, here's where it gets really interesting, because I have to push back a bit.
Okay, go for it.
Inserting 26,000 base pairs at 40% efficiency sounds incredibly powerful. But from a clinical perspective, doesn't that sound almost too powerful?
How do you mean?
Is this QuadPE system just wildly pasting massive chunks of DNA all over the human genome by accident? Because off-target effects accidentally causing a mutation somewhere else are the biggest nightmare in gene therapy.
You're absolutely right. It's the single most important question a researcher can ask. The scientists behind QuadPE were acutely aware of this risk, so they ran incredibly rigorous safety checks.
What kind of checks?
They use advanced sequencing techniques called GuideSeq and DiginomSeq.
Okay, so heavy-duty sequencing.
Yeah, these tools act like a fine-tooth comb, scanning the entire genome, specifically looking for accidental insertions or cuts.
And what did the fine-tooth comb find? Give me the evidence grading here.
High-rigor evidence. The off-target insertional events were essentially negligible. Furthermore, when they looked at predicted off-target sites, places where the DNA sequence looks confusingly similar to the target, the rate of unintended indels was incredibly rare.
How rare?
We are talking well below 1%.
And below 1%. I'm struggling to understand how it can be so safe when it's actively manipulating such enormous pieces of DNA.
It all comes back to the core mechanism we discussed earlier. QuadPE relies on a Cas9 nickase.
Right, a nickase.
Remember, standard CRISPR uses a Cas9 nuclease, which acts like a guillotine, chopping completely through both strands of the DNA helix.
The double-strand break that triggers the panic.
Exactly. A nickase, however, only snips one single strand. The DNA helix stays structurally intact. Because there is never a double-strand break, the cell never panics.
And without the panic.
Without that panic response, those wild, unintended off-target mutations simply don't happen.
Okay, time for recap number 3. QuadPE can paste massive 26 kilobase sequences. It is up to 61 times more efficient than previous integrase methods. And it boasts a near-zero off-target error rate because it completely avoids double-strand breaks.
Spot on.
So listener, knowing that the off-target error rate is virtually zero, how does that change your perspective on the safety of future genetic engineering? It's pretty wild to think about.
It really changes the math on risk versus reward.
Right, so we have a tool that is highly surgical, highly efficient, and works safely in a petri dish. But a petri dish in a lab is full of rapidly dividing cells.
Yes, usually HEK293T cells.
If we want to actually cure a human being, we have to deal with cells that absolutely refuse to divide. Does QuadPE actually work in the hardest environment possible?
This is where QuadPE truly separates itself from the pack and proves its clinical potential. The researchers set up the ultimate stress test.
What did they do?
They took K562 cells and chemically forced them to stop dividing. They used a drug called palbociclib to arrest the cells in the G1 phase of their life cycle.
Palbociclib.
And another drug called thymidine to arrest them in the S phase.
Going back to our analogy, they intentionally broke the car engine on the highway to see if the mechanic could still work.
Exactly. And as expected, when they tested traditional HDR editing, it completely crashed. It couldn't function without the cell actively dividing.
But QuadPE?
QuadPE completely ignored the cell cycle arrest. It maintained robust, high-level insertion efficiency, just as if the cells were still happily dividing in the dish.
Okay, proving it in chemically arrested cells is one thing, but did they test it in primary human cells? Tissues that naturally don't divide.
They did. To prove it works in real therapeutic targets, they tested QuadPE in primary mouse neurons.
Neurons, okay, that's huge.
Yeah, neurons are notoriously postmitotic. They essentially do not divide once they mature. QuadPE successfully inserted a massive 4.6 kilobase fragment into these resting neurons with up to 12.5% efficiency.
12.5%. That is massive for neurological diseases.
Yeah.
Conditions like Huntington's, certain types of Alzheimer's or ALS, the ability to drop a whole healthy gene into a living, non-dividing brain cell is revolutionary.
It really is.
But did they test any human cells?
They did. They tested it in primary human T cells.
Oh, T cells.
T cells are the frontline soldiers of our immune system and the absolute backbone of modern cancer immunotherapy, like CAR T cell therapy.
Right, where they upgrade your immune cells to hunt cancer.
Exactly. When targeting the TRAC locus in these human T cells, QuadPE achieved an astonishing insertion efficiency of up to 51%.
Wait, let's look at those numbers. 51% in T cells is incredible. Basically, half the cells took the massive upgrade.
It's unheard of.
But it only hit 12.5% in neurons. Both of them are non-dividing cells. Why the massive difference in efficiency?
It comes down to the physiological barriers of the specific cells. A primary human T cell, while it might be resting before an immune response, is highly dynamic.
It's ready for action.
Exactly. It's an immune soldier ready to mobilize, so its membrane and nucleus are slightly more permissive to taking in the cellular machinery.
And the neuron?
A neuron in the brain, however, is heavily armored. It is settled down for an 80-year lifespan. Its nuclear envelope, the fortress protecting its DNA, has incredibly tight pores.
Physical access is the problem.
Right. It is physically much harder to deliver massive 26 kilobase circular DNA chunks into a neuron's nucleus than it is to get them into a T cell.
That makes perfect sense. The biological environment matters just as much as the tool.
It does. And speaking of biology, I want to highlight a fascinating biological quirk about why the highly efficient PAM-out design works so seamlessly in these cells.
The one that duplicates the DNA, right?
Yes. I mentioned earlier that the PAM-out architecture generates a duplication of the original genomic sequence between the two nick sites. In genetics, we call these target site duplications, or TSDs.
Target site duplications, okay.
When you look back at natural evolutionary history, how ancient transposable elements, jumping genes and retroviruses, integrated themselves into the genomes of early mammals millions of years ago, they naturally generate the exact same target site duplication.
Wait, seriously, are you saying this brand new cutting-edge laboratory technology is essentially mimicking millions of years of ancient evolution?
Precisely. QuadPE is actively hijacking an ancient natural evolutionary mechanism for inserting large chunks of DNA.
Oh, wow.
Yeah, the cell's repair machinery happily recognizes and accommodates this PAM-out structure because, chemically speaking, it has seen it before in nature. It's an incredibly elegant example of biomimicry at the molecular level.
That is mind-blowing. Okay, our final mini recap before we close out. QuadPE works perfectly in non-dividing cells, achieving 12.5% efficiency in neurons and a massive 51% in human T cells.
Yeah.
And its mechanism mimics natural jumping genes by creating target site duplications.
That's right.
So with human T cells being the ultimate weapon in cancer immunotherapy, what does a 51% success rate in upgrading them mean for the future of personalized medicine? You know, it's huge.
It really opens up possibilities we've only dreamed of.
So what does this all mean? When we zoom out and look at this entire paper, QuadPE represents a true paradigm shift. We have moved past the era of messy, unpredictable double-strand breaks.
We absolutely have.
We have moved past the severe limitations of needing our target cells to actively divide. And we've completely bypassed the need to use those clunky, massive integrase proteins.
Good riddance to them.
Right. We now have a highly efficient nickase-based delivery system that can carry absolutely massive payloads up to 26,000 base pairs into almost any cell type with near zero off-target effects.
It's quite a list of achievements.
It turns a molecular typo corrector into a full-scale word processor capable of safely pasting entire chapters of healthy DNA into the human genome.
It is a remarkable achievement that changes the trajectory of gene therapy entirely. And I want to leave you, the listener, with a final thought to ponder as we wrap up today.
Let's hear it.
If we can safely and efficiently insert 26,000 base pairs of DNA into any cell dividing or not, without causing accidental mutations, we are no longer just talking about fixing broken genes.
What are we talking about, though?
We are crossing the threshold into synthetic biology. We now have the capacity to potentially install entirely new synthetic genetic circuits, think of them like specialized cellular apps, into human tissue to give our cells entirely new function.
Cellular apps?
Yeah. So I ask you, if you could upgrade your own biology, what is the first genetic app you would want to install in your own cells?
A profound and slightly wild question to end on. Thank you for joining us on this deep dive. Keep asking questions, keep challenging the boundaries of what's possible, and stay curious. We will catch you next time.